information shared on the Rethink It podcast is intended for educational and informational purposes only and should not be taken as medical advice. While Dr. Sande is a licensed medical provider, the content of this podcast is not a substitute for professional medical diagnosis, treatment, or advice specific to your individual health needs. Always consult with your healthcare provider before making any decisions related to your health or medical care.
Well, welcome back to the Rethink It podcast, you guys. I am so glad you're here. This podcast episode is one that you probably want to lean into, especially if you are anyone who is struggling from long COVID, have friends who are struggling, or family members. If you've been touched by long COVID, this is an episode you want to lean into. We talk about some of the the most recent research. We talk about how research is done. We talk about the process by which drugs are formed and the length of time that happens. There's a lot of great insight here to give you guys context. If you're not in the scientific world or the medicine world, you get some context into how we develop treatment therapies, at least in medicine. Dr. Ellis and I will have a great conversation. He is a medicinal chemist, meaning that he is a PhD, an associate professor in the academic world who looks for cellular targets for drugs. Okay. He looks to identify the way a disease process is operating, a mechanism to alter that process, and a drug and a target in order to create a drug to address that issue. So that's what a medicinal chemist does. Now, he has used his skills to basically do the opposite and instead of looking for drugs, he has using actual synthesized drugs, he has created a natural product that targets the micro-clot theory behind long COVID. So we talk about that in detail. It's a really great conversation. We talk about all of it. Just to wet your palate a little bit, after in the post show, and this wasn't included, so I have to include it here because you guys, I know you'll want to hear this. So prior to COVID, one of the researchers, now I'm blanking on the name he gave me, it was a well-known researcher in the thought behind long COVID being caused by these micro-clots in the bloodstream. This particular researcher had blood that had been collected from people prior to COVID. So no exposure to vaccines, no exposure to the wild virus of COVID. I know there's different thoughts on that, but just no post 2020 blood, just all pre 2020 blood. And he synthesized a form of the spike protein. This is after COVID. He's doing this research when people are developing long COVID, and he introduced it to that native blood that was blood from people prior to COVID. His theory was that it would cause clotting reactions. And we talk about how this happens on this podcast, so you're going to love to hear this. So he introduced that spike protein into the blood of these pre-COVID blood samples, and they all had a whole cascade of clotting occur. So it's a very interesting conversation. You're going to really walk away smarter. You're going to definitely feel a little bit more empowered and maybe even have a tool if you're struggling with long COVID. So Dr. Ellis is, like I said, a medicinal scientist, chemist. We spend a lot of time on some of that nerding out science. I promise I bring it in. I hope you guys are going to follow me just fine. I know you guys are smart, so I think you have no problem. But we do talk a little bit of chemistry there, and why this product, why triple therapy works, what the potential causes here in the micro-clot theory is with long COVID. We talk about the nicotine approach to treating long COVID. We talk about it all. So this is an all cast kind of conversation. We talk about everything when it relates to long COVID. And I think that you guys are really going to enjoy it. Now, we do also apply this theory, and we talk about how the implication of this theory and the science behind it can also impact dementia, Lyme disease, West Nile virus, all those post-viral reactive diseases that get lumped together, chronic fatigue syndrome and things like that. So there is something here for everyone, even if you are not dealing with long COVID. So I hope you guys lean in, take time to enjoy this podcast. It's a good one. Welcome, Keith, to the Rethink It podcast. I'm so glad you are here, and this is going to be a great conversation. I've already had a great conversation with you, so I'm excited to share it with the audience. This is definitely something that I know is still a robust thing. I keep hearing about long COVID and new different ways to approach long COVID, so I am excited about your product and the story behind it and like just the science. So without me dawdling on, why don't you go ahead and give me the backstory here, Keith? Tell me how you fell into starting a business, promoting a supplement product when you're a synthetic chemist.
Sure. And thanks for having me. I'm excited to be here with you today too. So I've spent most of my life as kind of a boring academic. I'm a medicinal chemist and organic chemist by training. I'm a professor.
Okay, all the scientists out there are over there shaking in their boots, all the medical people who had to take organic chemistry are like, oh, yeah, organic chemistry.
I do get that response a lot. And, and I do teach organic chemistry at this point. Yeah, so students, it's still hard for the students. So I'm a medicinal chemist. I'm a scientist. I've been a professor for about 16 years now. And so my academic career has been in my research. My academic career has been in medicinal chemistry. Mm-hmm. I kind of in that area in science where we think about as drug discovery, and medicinal chemistry is kind of the academic.
Sorry, say that again. That you consider as what?
Drug discovery.
Drug discovery. Okay, sorry. Dark.
So, I was like, medicinal chemistry is basically the academic discipline of drug discovery. Uh-huh. So, for instance, the students that I train in my lab, my graduate students, they go out and they get jobs, a lot of them in pharmaceutical industry, to do drug discovery and drug development.
Okay.
So
in the process, sorry to interrupt, but does it work where you do an academic research there? Then the pharmaceutical companies capture some of that academic research and they do their own development and then the hypothesis and test and all this stuff. Is that how that works? Just so that the audience, I don't think the average person knows how drugs are created, the process that we go through and the length of time that it takes.
Goodness.
And that kind of thing.
Let me tell you, the only thing worse than making a drug is when Congress makes a law.
Oh my gosh!
Ugly is the way that I would kind of start that, and wow. It's a long and it's a complex process. Mm-hmm. So, and it's also one that starts at a lot of different starting points, and then they all converge at the end. Mm-hmm. So, for an academic like me, I like to work on projects in the area that I think of as early lead discovery. So I like going into the literature. I like looking at new, what we call molecular targets. These are like the proteins that we're actually trying to modulate in the body to treat a disease. I like going in and starting projects on the very early end. It's just what I've always been interested in. So I will typically go into the literature, be looking at finding new molecular targets, new mechanisms of action to help treat a disease. And then a lot of times what I do is I'm looking for that initial molecule, what we call a hit. That is the first small molecule that we look at to start trying to develop a drug. And there's a whole process of drug discovery and development that starts with a molecular target and a hit. And goes to getting to a lead and lead optimization and then preclinical work. Where that work happens, there's multiple avenues where the drug it can be developed. A lot of that work can happen in an academic lab like mine, and then once enough data is developed and it looks promising, sometimes a company will come in and license it and take it on and actually develop the full drug. That's what happens when professors hit the jackpot, right?
Oh, okay. That sounds cool. And what university are you in? You're at?
I'm at Virginia Commonwealth University in Richmond, Virginia.
Right on. Okay.
There are great stories of faculty that have hit that jackpot and done some of that work. My mentor was someone that has advanced a drug into the clinic, and that's like kind of that aspirational thing that we're all trying to do.
Right.
So, but the reality is most professors and most academics won't do that. Most of them are contributing to a body of work in the scientific literature that maybe helps inform industry, and industry then picks up that work. And they do the industry tends to work more behind closed doors. They don't share.
Yeah.
They're interested in their intellectual property, right?
Sure.
That's how they make money on drugs. So
Sure.
a lot of that work happens internally in industry.
The
really hard part about the entire drug discovery and development process is it takes a very, very long time.
Yeah.
So from the point where you have a molecular target to start working with and developing and discovering it, that first hit, till the time a drug hits the market, is usually anywhere from 10 to 15 years.
Okay. Did you hear that? That is 10 to 15 years. That's how behind we are in treatment plans for diseases. I want you to take that into consideration when we're talking about context of COVID. Okay, continue.
Yes, and that's actually a big context to kind of what I'm working on now is it's this 10 to 15 year period from when we're actually ready to start working on that particular molecular target and using it to try and help treat a disease by developing a drug. Can it go faster? Yes, but the reality is the kind of the normal routine timeline we all think about and work with is that 10 to 15 year timeline to do all the work, including optimizing the actual drug molecule, doing all the safety testing, make sure it actually works. You start in a test tube and then you move to cells and then you have to move to mice and then you kind of ascend the various animal
biological data.
Get all that biological data, all that in vivo data, right? That data you have to get in a living animal, and you have to do multiple animal models for both what we call efficacy. Does the drug actually work the way you think it does, and actually help treat the disease state, as well as safety and toxicology, right? That you have to go through all that process, and then once all that's done, then you can start clinical trials, which is super expensive. Yes. And also takes a long time, right? Yeah. So it's not that anybody's like just slow rolling it or trying to go slow. It's just it is so much work and so much complex number of experiments and data you have to collect that it just takes time.
It just takes time.
Yeah. So in my academic work, I usually do early stage lead discovery, kind of that earlier work. It's much more mechanistic focused. It's kind of where the rubber meets the road,
right,
at a molecular level in biological systems.
Wow.
And that's always what's interested me in my academic career. And that was kind of my starting point for looking at long COVID. And then things went a very different direction.
Right. And what prompted the direction shift?
So in early 2023, COVID had been around for several years, right? Long COVID was starting to really be kind of the next problem on the horizon. And there was starting to be a lot of data in the literature. When you look at the scientific literature, if you just looked at like numbers of publications coming out, there's a huge number of publications in the COVID and long COVID areas being published, a lot of grant money. I said, well, okay, let me go into the literature. I want to see if I can find a long COVID project to get started, knowing there's this really long development timeline. And the sooner we get started on drug discovery, the better, right?
Right.
So I went into the literature to do the type of research I would normally do in the literature to find a good starting point. And after kind of doing the normal searches and looking at everything that I could, I kind of ended up at a place where there weren't really good molecular targets that were validated for me to start really working on and saying, okay, yes, this is something we can move forward and do the normal drug discovery process that we do both in academia and in industry, right? Because we follow the same process regardless of where we are.
Right.
And, it was one of those moments where you kind of lift your head up and you're a little bit shocked because it was one of those, it's how can we be doing so much research? How can so many papers getting published? How much grant money being spent?
Yeah.
And we are not making a kind of a focused effort to get to this starting point where we can really start creating interventions and therapies.
Yeah.
So it's that shocked feeling that makes you almost a little angry. It's how are we not doing
the basic
better than this?
Yeah. Basic. Yeah.
And it's really kind of a lack of focus. And it's not that there's not a ton of good work going on. And anybody in science knows we all stand on the shoulders of giants.
Mm-hmm.
Everybody's work is built upon the work of other people and the people
Right.
that came before them. And it's not taking away from anybody, just to kind of point out that the literature, while there is a lot of good work being done,
Yeah.
it's not being focused in the way that it needs to really develop therapeutics.
To get to the therapeutic, yeah.
So at that point, I had been talking to some people with long COVID. It was clear this was a real significant clinical problem.
Mm-hmm.
I had a good friend that had long COVID that came to me and said, Keith, I don't know what to do.
Mm-hmm.
He was getting passed around from doctor to doctor, from
Mm-hmm.
specialist to specialist, because
Mm-hmm.
nobody could figure out what was going on. And everybody was like, well, you have long COVID, but we don't know what to do for you.
Mm-hmm.
We just don't know.
Mm-hmm.
So he's getting no help. So that was when I was like, all right, well, I want to go back and I went back to the literature. I said, well, what can I do now? What is something different than what I would normally do, maybe, but
Mm-hmm.
is something that can be done now to actually try helping some people,
Mm-hmm.
right? Because that's, at the end of the day, everybody's in science and in medicine, in whatever other field they're in, to really try and help people.
Mm-hmm.
So I kind of just went back with a different mindset
Mm-hmm.
of what can actually be done to help people.
Yep.
And that's when I really started looking closely at a lot of the mechanistic data that was available. I kind of stumbled on the micro-clots hypothesis,
Mm-hmm.
which is a hypothesis about that micro-clots are one of the causative factors at a mechanistic level
Mm-hmm.
for long COVID, and really took a hard look at that hypothesis and the data behind it.
Mm-hmm.
And that's when I figured out, okay, here's something I can work with. I'm going to maybe have to do something a little different, but I
Mm-hmm.
can work with something and go in a direction that can help people.
Mm-hmm.
That was the turn, right?
Mm-hmm.
That was veering off the normal path. And I kind of just decided to go with it and see where it led. And it's
Mm-hmm.
been an interesting adventure.
So, and what did you discover so we had the mechanistic issue. So we knew that the theory was that micro-clots were contributing to the overall maybe anoxic and lack of oxygen to the brain type of symptoms. What was the standard treatment at the time, or was there any treatment at the time for that? Or were they looking at treatments?
So, and again, this was kind of a fascinating story. That was a fascinating question to ask. And the story in the literature is very interesting, because when you think micro-clot, you think, all right, well, let's use an anticoagulant,
Mm-hmm.
right?
Mm-hmm.
And
heparin,
so,
Lovenox, let's do it.
right?
Yeah.
So, and again, so you go in the literature, you ask that question, and people were aware of micro-clots. People tried using anticoagulants
Mm-hmm.
to try to address the micro-clots, but single anticoagulant therapy, like heparin alone, or some of the other
Plavix.
anticoagulants, exactly, those are the ones that people tried, they
Mm-hmm.
weren't effective against the micro-clot.
Hmm.
They didn't seem to make much of a difference just using an anticoagulant alone.
Mm-hmm.
And the interesting thing was then two of the investigators that are kind of some of the main researchers behind the micro-clot hypothesis, Riccia Pretorius and Doug Kell, they partnered with a clinician and did a small study. It was 90 people with
Mm-hmm.
long COVID,
Mm-hmm.
and they used a different approach. They actually used something called triple anticoagulant therapy.
Mm-hmm.
So it has actually 3 pharmaceutical drugs that they're all quote unquote anticoagulants.
Mm-hmm.
We'll talk about the mechanisms in a minute, but it's
Mhm.
basically 3 drugs. They did this kind of pilot experimental study on
Mm-hmm.
these 90 long COVID patients, and they actually measured micro-clots beforehand and after, right? So they had laboratory data on a mechanistic level to accompany what happened at the clinical level in terms
Mhm.
of symptoms.
Sure.
In that small study, they saw like an 80% recovery rate
80.
for people
80.
with long COVID.
80.
And it was across the board in terms of the symptoms. It was everything from the fatigue and the brain fog and the
Mhm.
cardiac symptoms to the mental health symptoms like the depression and the anxiety.
Mm-hmm.
It was across the board improvement in those types of symptoms in the
Mm.
people that it worked for.
Mm. Mm.
So that's one of those where it's like, it's a small study. It was a pilot study, right? It's been published as a preprint, because
Mhm.
I can only imagine why they haven't let that be peer reviewed published yet.
Mm-hmm.
There's all kinds of reasons you can think of,
Yeah.
some of which being legitimate, that it's
Mhm.
an underpowered study, that
Yeah.
there wasn't enough control. Like,
Yeah. Yeah.
but when you look at the data, especially when it's paired with the mechanistic data,
Right.
From doing the fluorescence microscopy assays to look at the micro-clots, you're like, okay, they're on to something.
Right.
So, and that was kind of a starting point where I was like, all right, well, why isn't everybody just getting this? Right. And I went in and talked to some of the long COVID people.
Just for curiosity, what were the 3? I think I saw the study, but I filed it because I'm like, I'm not prescribing.
Right.
What were the 3?
The 3 drugs in there, it's apixaban, which is a factor 10A inhibitor. That's a true anticoagulant. It was low-dose aspirin, so like the 81 milligram. That's a COX-1-2 inhibitor, right? Technically, that's a platelet activation inhibitor. And then clopidogrel, which is a P2Y1, P2Y12 receptor antagonist, which is also really technically considered an anticoagulant. Or sorry, technically it's a platelet activation inhibitor, but everybody treats it as an anticoagulant because those two kind of have the same effect.
And I'm just curious. I think I vaguely remember, but just for the sake of my audience, because I'm just forgetting some of it. And when you take an aspirin, how long does it activate platelet inhibition? Is it like 7 days, 10 days? I feel like it's I don't remember
that. I don't remember that.
That's a surgery question. So I'm thinking if I took triple therapy, Plavix, I think it's 48 hours off of Plavix and 7 days off of aspirin or something, because it's like a permanent
right, because aspirin's an irreversible inhibitor, right? So you have to actually clear it all out of your system before surgery to make sure you get rid of that effect. I don't remember what the half-life of it is. Man, that's
not really that important. I just keep just
no, it's actually a really good one. I never even thought about, but that one is a little long acting for that.
Which is, by the way, why your doctor gives you a low-dose aspirin if you're at high risk for a heart attack. So those are the 3. And so how did we get from those triple therapy pharmaceutical level to your product? What's the name of your product?
So the product I developed is called Circuguard.
Oh, Circuguard.
Basically, when I was talking to the long COVID patients, I was asking them, I was like, well, triple anticoagulant therapy seems like it's pretty good. Why aren't you guys all taking it? Right? And most of them couldn't find a doctor to prescribe it. So with that, I kind of took that piece of information. I stepped back a second and I was like, all right, well, what's going on here? Why would that be? So, and there's actually multiple issues of why triple anticoagulant therapy is somewhat hard to get, right? So the first is you're taking 3 anticoagulants together. There's a significant safety issue there, right? There's a high risk of bleeding as a side effect. It's not something you're going to give people without some real careful consideration.
Good explanation.
Right.
And you got to be able to defend it to the medical board, man.
Exactly. So that alone is actually a reasonable, reason why that alone it is a reasonable criteria for not giving it to people is okay. There's a safety risk. So the other reason I kept hearing from people is a lot of doctors didn't want to prescribe it because there wasn't adequate evidence-based data, right? There was no randomized placebo-controlled clinical study showing that it worked. And it's like, well, that gets to be a chicken and egg problem, right? If you're not going to give it to people, you're never going to figure out it works.
13 years for development of evidence-based medicine studies,
Right. And clinical trials take time, right? So if you want to wait for a clinical trial of triple anticoagulant therapy for long COVID, it's probably at least 3 to 5 years minimum to get that trial done, get the data analyzed, and actually publish it and create guidelines for physicians to prescribe it, right? So that was kind of strike 2. So it's got a safety issue. There's kind of an access issue, right? Because the physicians don't want to prescribe it. And then there was a cost issue because these are not cheap drugs, especially the apixaban and the clopidogrel are not cheap drugs, and insurance companies don't necessarily want to cover them.
Well, they're not only not
used off label.
Yeah,
right?
definitely not going to cover it off label. I mean, I can't tell you how many hours I spent on the phone with an insurance company defending my prescribing of a certain drug because there was a different one that I should have given that was cheaper. And this is maybe off-label. Yeah.
So
Off-label.
what I was hearing from long COVID patients is, a lot of these private pay long COVID clinics have popped up across the country. That's one of the few places you can get micro-clot testing done because there is no widely available clinical test for micro-clots, right? And this is the frustrating thing, I think, for a lot of long COVID patients is physicians will run all their normal blood work panels and the person looks fine.
Yep.
But they are hardly functional. They can't get out of bed and all the other symptoms that they have.
Yep. Your labs are perfect. It's like the meme of the guy in the casket and the doctor said, but your labs are fine.
Right. Exactly. So from the doctor's point of view, it's like, well, just go home and rest. And the long COVID patients are like, are you crazy? I'm sick. Like, help me.
Yeah.
So there is a research lab-based assay for micro-clots. Some of these private pay long COVID clinics are actually running that assay for people. People can get their blood checked to see if they have micro-clots. Some of those clinics are actually prescribing triple anticoagulant therapy. There's still an access and a cost issue, right? Because those clinics, most of them have long waiting lists. You have to wait to get into them. And they're private pay. A lot of them don't take insurance at all. So there's still a significant cost barrier there. So at the end of the day, kind of looking at the whole situation of what was going on with triple anticoagulant therapy and long COVID, it's like, well, people kind of know that there's a reasonable hypothesis and some reasonable data that this works. And what you really need is a safer, and the way you make it safer is by making it less potent, right? So there's less of a bleeding risk.
Right.
You need a safer, accessible, without a physician, and inexpensive version of triple anticoagulant therapy.
Right.
So, and this is where I kind of did something that is really radically different than what most medicinal chemists would do. So understanding that was the situation, my original kind of training, my PhD was in natural products. It was in natural product total synthesis. And then my training as a postdoc in medchem was in using natural products as the starting point to develop new drugs. My mentor mainly worked in oncology and antibacterials, and that was where I started in my own independent career. There's a long history.
So I just want to emphasize, you were starting with a natural therapy in order to develop a drug. So you had the natural therapy that you knew did something, and from the molecular structure of that natural therapy, you were developing a hypothesis to synthesize a drug. Is that right? And make it more potent.
Yes.
Just want to make a point so people are understanding this is where we do some of this, drug development often starts with what we see in nature, just in a synthesizing and more potent, potentializing, making it more potent way. I just think that's really important.
Up until about the year 2000, that's the way we developed most drugs, right? And that paradigm and the model we use for drug discovery has shifted significantly over the last 20 years. But the real old school way was you started with a natural product, you figured out what about the natural product gave you the biological activity you wanted, and then you used all that information to create your drug.
Isn't that amazing?
And that's
an argument for natural therapy.
It really is, right?
Yeah, it is.
And that's how I started my career. So I said, well, in this case, we know this pharmaceutical therapy kind of works for this, but it's too powerful. The drugs are too good at what they do. We need something that's safer, and that in being safer is something that's more accessible for people, because if there's a lower safety risk, it's something people can take on their own.
Right.
So basically, I kind of flipped the script of what I was always taught to do with using natural products as the starting point for drugs. And I said, well, let's take each of these drugs in triple anticoagulant therapy, and I basically reverse engineered triple anticoagulant therapy using what we call the mechanisms of action. So the mechanism of each of the pharmaceutical
How do these drugs work?
Right.
That's mechanism of action. It's fancy. Yeah. We have all these fancy terms like pharmacokinetics and pharmacology.
That's right.
Mechanism of action. It's just, how does it work?
So I started with the natural product, started with the pharmaceutical drug, looked at the mechanism of action. I said, okay, well, what are natural products or natural ingredients that have the same mechanism of action? And I went and found those and said, okay, can we custom design what I think of and what I call a natural version of triple anticoagulant therapy? And that's actually the product that I created what I call Circuguard, right?
Mm-hmm.
So as I was doing all of this work, I was publishing kind of the research I was doing on a Substack, because it was a good way to reference all the literature I was looking at. And basically, I went and did all of the work and said, okay, let's reverse engineer the pharmaceutical drug to a natural ingredient, worked through, figured out all the natural ingredients I wanted to put in this natural version of triple anticoagulant therapy. And at that point, I said, well, okay, great. Well, now how are people really going to take this, right? How is it actually going to help somebody? Because what I had was information, right? So, and that's the point where I really kept going down the pathway and a departure from the normal. And I actually went ahead and formed a company that is in effect a supplements company, right? And had the product manufactured. And one of the reasons I really wanted to do that was to make sure it was using high quality ingredients, to make sure it was being manufactured under proper conditions, under GMP conditions, that it was being.
I don't think anybody's more qualified for that than you are.
Right. It was kind of a natural thing to do. It's like, well, who else is going to know better about quality control and getting that third party testing done. So I went that step of actually having the product made, and people that were kind of following my work and that I had been talking to that had long COVID, they started taking it, and it really seemed to help a lot of them. And it would help. The way I describe it is it helps make them more functional in their own life. What people tell me is it helps with the brain fog. It helps with the fatigue. It helps with some of the cardiac symptoms. I've actually had a couple people go as far as telling me that it's helped with their dysautonomia, which is dysfunction in the automatic nervous system, right?
These people getting up, passing out.
Right, and it helps with some of the neuropathy issues and being able to regulate the breathing and the heart rate. So it seems to really be helping people. And does it completely get rid of all the long COVID symptoms and cure them? No, it's a supplement, right? That's not what it's meant to do. But it's also a safe and natural remedy. That it doesn't have a huge risk of side effects for bleeding. People can get their hands on it. They can order it and try it if they think it's going to be something that might help them. And it's fairly inexpensive. So it checked all the boxes of giving people something that they could use to help their long COVID, that they could actually have the freedom to choose to actually try for themselves without anybody else's permission, and that would be safe no matter what if they took it.
Right. So what ingredients have you got in there in Circuguard?
So there's organic turmeric. The active ingredient in there is curcumin. And curcumin's actually a factor 10A inhibitor, which is the mechanism of action for Pixaban, right? So that's the anticoagulant. There's willow bark extract, which is the kind of a natural version of aspirin. It's the COX-1 and 2 inhibitor. That's the plate activation inhibitor. And then the replacement for the clopidogrel, which is the P2Y1, P2Y12 receptor antagonist, the replacement for that is a product from traditional Chinese medicine called Danshen. It's an extract. There's a really nice scientific reports paper that shows that there's two natural product components in there. Sulfonic acid A and B are both P2Y1 and P2Y12 receptor antagonists.
Wow.
So those are the three main components. Those are the three. And what I tell people is it's not about any one mechanism of action. It seems to be about taking all three together, whether you're taking the pharmaceutical version or whether you're taking the natural version. You have to use all three of those mechanisms of action together to actually treat
because the studies prior
micro-clots.
to with just the single therapies were not successful. They showed no outcomes.
That's right.
Yeah, that's really interesting. As we're talking about long COVID and micro-clots, we know, and this is a brain health and longevity podcast, we know that theory of micro-clots in the nervous system is also something we look at and consider and review and look for in early dementia. It is known that micro-clotting in certain areas of the vascular system of the brain leads to dementia. We know that, and we look for that on scans. There are other things that we could say micro-clotting in the nervous system causes inflammation of the nervous system, can be contributed to a whole host of neurologic issues. So it's super important, even though this is targeted toward a long COVID picture. I feel like this with the combination of ingredients that you have there, I could see this being a valuable product in the space of just brain health and longevity when we're looking at a picture of somebody that's very potentially has micro-clotting issues in the nervous system. Thoughts on that?
No, well, I think that's the whole micro-clot role in disease states, as I think a really huge area. It's got a real potential to help a lot of people, because the way that I'm looking at it, because it's not just dementia. There's some other disease states: Lyme disease, CFS, chronic fatigue syndrome, West Nile. There's even data showing that micro-clots occur in diabetes, type 2 diabetes.
Oh yeah.
It's really interesting, because it's like you've got all these disease states and everything but the diabetes and the dementia. But if you look at long COVID, chronic fatigue syndrome, West Nile, and Lyme, they're being grouped in what's called, this set of diseases called post-viral reactivation diseases, right?
Yeah.
And there's starting to be data, like I think there's some data that micro-clots happen in chronic fatigue syndrome.
Sure.
So it appears to me, I think what's happening is at a symptomology level, at the symptoms level, chronic fatigue syndrome, long COVID, West Nile, Lyme, and even dementia, right, kind of have these overlapping constellation of symptoms, right?
Sure
that
do.
all seem to be downstream of the micro-clot. And it's almost like all of these diseases have in common that micro-clots are being formed. I think at the molecular level, what we're gonna find, I think if somebody eventually does all the studies that need to be done, I think what will happen is, what we're gonna see is that at the molecular level, there is a different, like, direct cause of the micro-clot formation. So in long COVID, and there's really good data on this from the team micro-clots and the micro-clot hypothesis, there's really good data showing that the spike protein binds to the protein fibrinogen in blood, which was the main protein that's used for clotting. Basically, spike protein binds to fibrinogen and causes an unnatural conformation shift in fibrinogen, and then
making it
polymerizes.
hypercoagulable. So that
yes, and that's what the micro-clot forms out of, is this incorrectly folded version of fibrinogen. And in a lot of the early micro-clot papers in long COVID, they refer to them as amyloids.
Interesting.
They're amyloids of fibrinogen.
Amyloids. That sounds awful familiar.
I thought you would like that word, right?
Yeah.
Where we think about Alzheimer's as
amyloid plaque
amyloids
formation,
of tau or A beta, right? So I think what we're going to find in a lot of these disease states where micro-clots are being implicated is at the very basic, most molecular
level,
zoomed in picture. You're going to see a different protein is initiating the formation of this amyloid micro-clot. But that the amyloid micro-clots, when they're disordered and they form into these larger aggregate structures, they're behaving the same in most of these disease states, which is why they're causing similar but different symptoms. That from a different root cause in a different physiological system, it's giving you kind of the same symptoms.
Similar constellation of symptoms.
Yes. So I think that's probably what we're going to find long term when we do all the work. Now, cutting to the, okay, well,
it's
how
interesting
do you help people?
Because the data prior to COVID, and I cannot find, I dig, and I cannot find data after COVID. But prior to COVID, we were seeing this in massive spike in early onset dementia.
Yes.
Like a 400-fold increase in like 4 years prior to COVID. And now we consider our long COVID in this picture. I'd love to see the data. I'm putting it out there publicly. I want them to give me the dang data because I cannot find it. But I suspect that it's triple what it was prior to, which is terrifying because early onset dementia is an age younger than 40, or younger than 50. Sorry, that's dementia in your 40s.
Well, and it's interesting to see, is some of that uptick in dementia, is it getting lumped in with other things, right? Where it's not being recognized as just on its own now, because there's this kind of complexity now in the system of all of these disease states that especially the physicians don't quite know how to deal with. They don't have solutions for. There is no evidence-based algorithm.
For Lyme, long-term Lyme, or chronic fatigue. It's just we give some antidepressants, right?
Right.
Which, by the way, that's given me a little segue. I don't mean to interrupt, but we give antidepressant, pro-serotonin drug, antidepressant to a person with chronic fatigue who might have micro-clots. How does that affect the clotting?
So it depends on exactly what the agent is and what it does. You had mentioned 5-HTP, right? That's the precursor for serotonin. So there was a really good paper out late last year that showed that their patients had compromised, long COVID patients had compromised serotonin uptake from the gut, right? So a lot of people started loading up on 5-HTP, which is the precursor for serotonin, they need to try and absorb more of it. What I haven't seen come out is whether, like in long COVID, it is the receptor for 5-HTP that is responsible for uptake in the GI tract. Is that compromised as well? Right? So there's this kind of supply and demand issue with serotonin. It's like, are you able to uptake enough serotonin directly or 5-HTP to make more? And then interesting, the serotonin tie-in with long COVID is actually to the platelets.
That's my question. Yeah.
Right? Because
now this is fascinating to me because, I mean, my understanding of serotonin from the time I went to medical school or PA school to now has just developed and developed and developed. And it's just crazy to me because it's only, in school, you're like, this is your happy hormone. We need to give serotonin drugs to make you not depressed. That's where we start. But then we find out, serotonin regulates, heart rate, blood pressure. Wait a minute, it comes from the gut. And hold on, there's more. It affects the platelets. So tell me about that.
So one of the reasons it affects heart rate and blood pressure is because one of the major storage locations for serotonin in the body is in the platelets.
Wow.
Right?
I did not know that.
So what happens is in long COVID, when micro-clots develop, so we talked about these amyloids of fibrinogen. You get these like sticky, unnatural amyloids, and they also stick to platelets and red blood cells. So when they stick to red blood cells, all of a sudden you're impairing oxygen delivery to the tissues. You're getting hypoxia, which is what's causing the fatigue and brain fog, neuroinflammation. And when the platelets stick to the micro-clots, you're getting what's called platelet hyperactivation. They're going into overactive mode. When that happens, they dump all their serotonin. So it leads to serotonin depletion in the body. Which channels
But also that's an autonomia that you're going to get. You're going to get all this.
It's autonomy, and it kind of rolls downstream symptom-wise to depression and anxiety. Right? So because you're compromising the serotonin storage system by causing a constant state of platelet hyperactivation.
Oh my gosh. Wow.
Right? So, and this is why long COVID is really such a bad disease, and why I actually feel a lot of sympathy for the physicians. I feel like I kind of beat up on them sometimes for not helping patients enough, but it's such a complex disease. Long COVID is this multi-system disease, and the physicians are really taught to deal with things system by system
and symptom by symptom. So we take our physiology and our organic chemistry and our biochemistry prior to medical school. And then we go, thank God we're done. Seriously. Now let's do diseases and drugs. Okay, that's seriously. And I was shocked when I took a naturopath or, got my PhD in natural medicine, realizing that everything I do on this space is based on biochemistry that I learned prior to PA school, right? So you're like, okay, wow, I shouldn't have dumped that information. I should have maybe learned it a little better.
So when people with long COVID are walking into a primary care physician's office, they have
mitochondrial dysfunction
issues. They've got mitochondrial dysfunction. They've got cardiac symptoms. They've got fatigue. They've got neuroinflammation and CNS symptoms. So the primary care is like, okay, I'm referring you to the cardiologist and the cardiologist, and let's send you to the rheumatologist and $1 million
workup.
That's why people are getting passed around, because long COVID it's hitting all of these different systems causing all these different symptoms. But at a molecular level, no one's really been addressing yet why that's happened. And that's why I've become a big believer in the micro-clot hypothesis, because it seems to be almost like a unifying theory behind what's going on physiologically, causing the symptoms in multiple systems. It's actually a rationale that explains just about all of them.
Yeah, that's incredible. That, and that I would marry that with cell danger response from the mitochondria. So those two, I think, would be your two big issues.
Well, and we haven't talked about involvement of the immune system at all, right? Which is obviously compromised.
That's cell danger response.
With
Let's talk about that.
Yep.
So what happens?
I'm not familiar with cell danger response.
So that's the work by Richard Navio out of University California, San Francisco. So I would recommend you do look into some of that research by him. As an academic, I think he's in as well. I think he's a biochemist maybe, but he's the one who coined it. And cell danger response is a way he describes the immune component of chronic disease, where the mitochondria assume the role of an immune responder rather than the battery of your cells. So you've got your mitochondria, whose primary function is a voltage reader and then energy producer, right? But there's also this function it has of when there's a voltage shift, like let's just say the flu, the mitochondria exit the cell, they act as immune modulator, immune responder. That's why we get so severe fatigue when we're having the flu. And the idea is to respond, help the immune system respond, and then adapt. It's a normal response. It's a healthy response. Create more adaptive cells with healthier mitochondria able to respond to that future stress. But the problem is with cell danger response in a chronic infection or chronic disease state is that the mitochondria exit and they never re-enter. And so you end up with chronic fatigue, or dysfunction of systems where the organs don't work well.
Sure.
Whichever the organ is.
And that makes complete sense, and especially that it would happen in really any of these post-viral reactivation diseases, because the part of the central part of that hypothesis is there is some sort of viral persistence in all of these. Like, so long COVID, at the end of the day, long COVID, you've got to have a source of spike protein from somewhere. There's like these viral reservoirs in the body. There's a lot of debate as to where they are. Are they in the gut? Which is actually kind of a pretty good place. It's a lot of people think it's what's responsible for the gut dysbiosis that happens in long COVID patients, is the gut's acting as a viral reservoir, and then you're getting transport in slowly over time. So there's like this persistent low-level viral infection that would touch that off and cause that mitochondrial response chronically over time, and you're never going to recover from.
Yeah, exactly. Yeah, those are very interesting. I'm just curious because I've heard. I've read a little bit about the most recent thing in long COVID or treatment protocol that people have touted as a thing is nicotine.
Yes.
Have you heard? What is your thoughts? Can you speak on that? I know it's weird.
I see a lot of people try it. It seems to be hit or miss. Some people swear by it. It's
pretty high dose nicotine, by the way. And patches,
it is.
Not smoking.
Yeah, it is the patches. It is kind of high dose. I think, for some people that don't have, that I could see where it would be a problem for people if you had nicotine issues in the past, but if you've never smoked, you've never had any nicotine addiction issues, you slap it on and try it and see what happens. I don't see any problem with it. I have never seen good molecular level data though, as
that's
to
why
what
I wanted
the
to
mechanism is supposed to be. And maybe it's out there and I haven't seen it, but I haven't seen any kind of commonly accepted mechanism behind what the nicotine is doing.
That's what I wanted to hear. Because ultimately, we're trying to be as scientific as possible, even in the natural health space. Like, we have to understand what it is we're trying to achieve before we decide we're going to give people a series of supplements. Like, when I talk to clients, I say, this is the plan. Here are your supplements. This one's doing this. This one's doing this. This one's doing this. The goal is this, and that's a scientific mindset, right? That's still reason behind that, mechanism behind that, and all that.
And that's one of the reasons I had a real hard time internally, like myself, kind of really pushing forward and actually formulating it and making my supplement. Because I was like, well, is this really the best way to help people? But when you look at what people are taking for long COVID, a lot of people are taking stuff without real good mechanistic rationales. It's a lot of
throwing spaghetti at the wall.
That's right. And even in some of the clinical studies, there's kind of not necessarily good mechanistic rationales. They're trying to repurpose a lot of things, whether it's a pharmaceutical or a nutraceutical, right? A supplement. There's a lot of repurposing going on where it's not clear to me what the mechanistic rationale is behind the repurposing. And one of the reasons I really pushed forward and did what I did was I was at least custom formulating something based on mechanisms of action. Like scientifically, that's something
you're still
I
walking
can feel
scientifically.
good about doing.
Yeah. You didn't leave your ethics behind.
That's right. And it's something that I feel like has a good hypothesis behind it for people to try. And is it going to help everybody? No. But even pharmaceutical drugs don't help everybody.
No,
right?
In fact, I think that if we have a 50% recovery, we think we're doing great with pharmaceutical drugs.
So, but yeah, a lot of the stuff that's floating around, and I haven't had a chance to look at it. I saw a brief social media post on a study that came out just, I think, this week on repurposing both pharmaceuticals and nutraceuticals for long COVID. And the horrible part was like the response rates were, for whether they helped, I don't think any of them helped more than 50% of the patients.
Yeah.
Right. So you're getting very low response rates, low actual improvement in symptoms and functionality, even when you are getting what these successful ones that are being repurposed are not even doing that great a job for the people that they're helping.
Right. Well, what I like about the formulation that you made and the product line is that it's relatively simple. Like when you think of like the nicotine protocols that I've read and heard, they're not easy. They're complicated. There's a titration period. It's do this and then wait and then do that. It's just complicated. Whereas this product seems pretty straightforward and easy. And
it is. And that
any contraindications?
If you're already taking an anticoagulant, right? So if you're already on an anticoagulant because your doctor put it on for your long COVID, maybe not take that.
Yeah.
But other than that, most people, or if they have some kind of allergy, like if they have an aspirin allergy, I wouldn't take it. But most people know if they have an allergy to things like turmeric or I haven't heard of any real allergy issues or adverse events for Dan Shen. I also put some beetroot extract in there because nitric oxide helps with oxygenation to help with some of the fatigue recovery. I have heard some people say that there is a real rare like beetroot root vegetable type allergy.
That's so rare. It's got to be so rare.
I had never heard of that one when somebody mentioned that to me. So there's not really a lot of contraindications. My goal was to make it really clean because a lot of people with long COVID have a lot of immunological issues going on.
Sure.
A lot of them have MCAS.
Yeah, boy,
of sensitivities.
Oh, yeah.
So when I was making it, I wanted it very clean. It's got the three components
low
it's histamine in it. Yep.
Tolerance response type thing.
That's right. It's in veggie caps to not cause additional MCAS type activation. And it's really stripped down. It's got some black pepper extract in there, a very small amount that helps with the bioavailability of the turmeric. So, but other than that, that's all that's in there. And I wanted it simple. So it's 2 capsules is the standard dose. I did have, when it first came out, I had a lot of people ask me, they're like, can I take more? Like, I want to get better faster. Can I take more? And I actually went through and I looked up like the max recommended dose for each individual ingredient. And it's way below that. So I was telling people, if you wanted to double up, you're probably good. I think I told people don't go beyond like doubling up. But even if they did, it's still super safe because like for turmeric, there's how much you say? 400 milligrams of turmeric in here. The max doses, if you go on Amazon, you can buy like 2,500 milligrams turmeric capsules. Like
you could have diarrhea for a week.
Right.
Like, my gosh.
But the real reason that Circuguard works is because it's the combination of the ingredients together. And yes, if you take more, you might get better faster. And depending on how severe your long COVID is. Because a lot of people will also ask me, how long do I need to take it? with the pharmaceutical version of triple anticoagulant therapy, they kind of segregated the people into two groups. The mild long COVID patients were on the pharmaceutical version for 2 to 4 months. The severe were on it for 4 to 6 months. So I basically tell people, depending on how severe your long COVID is, plan on taking it for 3 to 6 months.
Yeah. So don't do a one and done. Give it a minute. Natural therapies are like that. As a rule, I just would put that out there. We don't do something for just a week, or don't expect it to work like a drug. Like, you're going to need to do, especially for something chronic. I mean, granted, I do have some products, work great for inflammation today, but if we're trying to go for something chronic like long COVID, then we want,
And also for this with micro-clots, what you're really doing is it's helping support the body naturally clear the micro-clots. Right. So by, it's a natural version of triple anticoagulant therapy. What both the pharmaceutical and the natural version are doing is they are throttling back and decreasing the amount of coagulation going on. It's slowing down clot formation so that new microclots aren't forming, and it lets the body naturally clear the microclots that are already in the bloodstream. And that's where the pharmacological effect is coming from.
I'm just curious, and I can't remember how long platelets last in the body. Do you know? I can't remember.
No, that is deeper into anatomy and physiology than I usually go.
That's the stuff that I like to know. Okay, let's take a look. 10 days. So that is why aspirin is 7 to 10 days. We have to have a whole new set of platelets.
Platelet synthesis. Yeah,
that makes sense.
And that makes sense.
There you go. Sorry, I totally nerded out there for a second. I had to know right then. Awesome. Well, I have really enjoyed, in so many ways, this conversation. I've learned a lot. You really challenged my understanding of long COVID from the micro-clot perspective. I did understand that theory, but I have a better understanding of it now thanks to this conversation. So thank you for that. And I really loved all the research, your background, and I hope that this product really does take off for people, because there's so little available when it comes to resources in COVID, long COVID. So I'm thankful.
And at the end of the day, that's really why I did what I did. It's very different than what I've ever done the rest of my career, but nobody's really helping people with long COVID that much. And this was something that I thought would actually really help people and at least give them a choice and a tool to try to help themselves recover.
Yeah.
Because the long COVID community, I have to say, they're very resilient.
Yeah.
And they've become very self-reliant.
Yes, because nobody's helping them.
Get help from anywhere else.
Yeah.
Right.
Yeah. Yeah.
So I kind of felt like, with my background and knowing what I know, that this was one of the best ways that I could help people with long COVID and help them now and not help them in 10 or 15 years.
Right.
Which is the way the timeline the rest of the world is operating on.
Right. That's crazy to me. I mean, it's just crazy, but it is the reality. It's always been the reality just because of the way research goes. So that means that our most advanced drugs are 15 years old, potentially. Research is.
Much. Yeah.
So cutting edge medicine. Yeah, I tell you what, it's interesting because the mindset behind the triple therapy, and I haven't used your product, but I've used those ingredients in combination inadvertently.
Right.
In my population with long COVID, which thankfully I don't have a huge population, and I deal a little more with the Lyme community and some of the brain chronic fatigue, but that seems to be an overlap, right?
It does, and it's one of those. I'm really waiting. I know there's some people working on like micro-clots and chronic fatigue syndrome. I would love to be able to see if this combination or maybe a minor alteration of it would be able to help with micro-clots in some of these other indications, some of these other disease states where micro-clots seem to be a causative pathophysiology. It's one of those, I'm waiting for the research to hopefully catch up.
That's so cool. I'm so thankful that you came on. So where can our audience find your product? Where can they find you if they have questions and those type of things? Leave us with that.
The best place for people to go is my website. It's KeithCellisPhD.com.
Okay.
And that's kind of my page of links, right? So there's a link to my Substack. If you want to go read about all the research and how the product was developed and all about micro-clots, kind of general audience level writing, that's all on my Substack. My social media is there. there's a link to go be able to buy Circuguard. There's a link to the product page there that gives you all of the detailed, nerdy, scientific information on what it is and why it works.
So cool.
And if they go there, they can reach out. They can email me. My LinkedIn profile's there if people want to check me out, figure out whether I really am a scientist and really am who I am, because I understand people's natural skepticism.
Oh yeah, especially
especially in the long COVID community, they feel like they've been taken advantage of by people. So I try to be very open and transparent and put it all out there, and people can look at it and make their own decision. It really, I've used the principles that I've been using for my whole 16 years as a professor and all of my time as a scientist before that to do this stuff. So that's kind of what the knowledge and expertise that's gone into really trying to put this together.
Amen. That's so great. Awesome. Well, I will include that link also in the show notes and, of course, in the email that goes out to our audience that is subscribed to us. And I'm so thankful again that you came on today. All right.
I appreciate it. Thank you for having me.
Yep. I'll talk to you soon.